Hey, I'm Dylan Wallis, and welcome to my poster. Today I'm going to be talking about using a high-throughput screening assay to identify genetic mechanisms of individual susceptibility differences in a genetically diverse population of zebrafish. Differences in individual susceptibility to chemical exposures have important implications in environmental health sciences and toxicology. Understanding the mechanisms that cause these differences and chemicals that can participate in large differences in susceptibility could help protect vulnerable populations, help to better regulate chemicals, and could lead to better individualized medicines. The high-throughput screening assay exposed thousands of zebrafish from a genetically diverse zebrafish population to thousands of chemicals from the ToxCast chemical dataset. Of these chemicals, abamectin was selected for validation studies because it showed high variance across multiple endpoints and multiple doses in the screening. A preliminary genome-wide association study was done to find regions of the zebrafish genome that were associated with susceptibility to abamectin. A region upstream of the gene SOX7 was identified, and a deep reanalysis of that region was done, which identified several SNPs and an indel of varying length, which were associated with differences in susceptibility to abamectin exposure. The top portion of Figure 1 shows a multiple sequence alignment with representative sequences in that region that show the main SNP that was associated with abamectin susceptibility, as well as two different length indels that were also associated with susceptibility to abamectin. Real-time PCR experiments show that there were significant differences in SOX7 expression between fish that were extremely affected by abamectin exposure and fish that were unaffected completely by abamectin exposure. This two different sequences that were associated with the fish being either extremely affected by abamectin exposure or completely unaffected were cloned upstream of a luciferase expression vector and transfected into NCF7 cells. These cells were then exposed to abamectin, and expression was measured using a luciferase assay. The results of this experiment can be seen in Figure 2, which shows a clear difference in luciferase expression between both sequences when they are exposed to abamectin, as well as differences in the controls. It also shows a large difference in luciferase expression between both inserts and the vector without any promoter insert at all. These results indicate that there is some sort of regulation differences occurring based upon the region that we discovered upstream of SOX7. Our hypothesis is that these results are caused by differential transcription factor binding at the sequence upstream of SOX7, which leads to differences in SOX7 expression, which cause differences in susceptibility when exposed to abamectin. To test this hypothesis, transcription factor binding site prediction software, MatBase, of the Genomatics software suite was used to try and predict transcription factor binding site differences between these sequences. At the bottom of Figure 1, you can see some examples of transcription factor binding site prediction on our sequence, lined up with the multiple sequence alignment showing examples of the sequence to get an idea of what this looks like. After transcription factor binding site prediction was done, these sequences were compared using a chi-squared test to see if there were any significant differences between transcription factors that were bound to either sequence. The results of this can be seen in Figure 3, which shows some highly significant transcription factor binding site differences between the two sequences, some of which are located inside of the highly variable region that were discovered in the GWAS experiment. Guided by these computational and wet lab results, future studies such as IMSA and ChIP-seq will be used to verify transcription factor differences across sequence that are causing susceptibility differences to abamectin exposure. This study validated a high-throughput screening technique that can flag multiple chemicals for signatures of individual susceptibility differences across a genetically diverse population. It also showed that this screening identifies signatures of gene-environment interactions that can be probed to identify mechanisms of individual susceptibility differences. These results have far-reaching consequences in chemical regulation and individualized medicine that could result in more diverse and equitable systems to design drugs and regulate chemicals quickly and effectively. Thank you for listening to my poster presentation and have a wonderful day.